Hello everyone, welcome to today's live broadcast, Producing Petri Puppies, Gamete Collection and In Vitro Fertilization in Domestic Dog. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars, contributing to the advancement of science through content sharing capabilities. LabRoots is a powerful advocate in amplifying global networks and communities. Before we get started, I have a few important announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We're going to try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of your slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button, lower left. I'd like to introduce today's speaker, Dr. Jennifer Nagashima. Dr. Nagashima is a Smithsonian Institute postdoctoral fellow for the Smithsonian Conservation Biology Institute. Her current research focuses on developing new gamete rescue technologies, and candids and felids include freezing sperm and culturing ovaries in vitro to grow mature eggs. As part of her doctoral dissertation, Nagashima produced the world's first litter of puppies from in vitro fertilization and cultured domestic dog ovarian follicles using cutting-edge three-dimensional systems with the goal of applying these technologies to the conservation of wild candids in the future. I'll now turn it over to the doctor for her presentation.
Hello everyone, thank you so much for joining me this morning, and thank you so much to LabRoots and to Robert for that introduction. So I'm going to be talking to you a little about the work that I've been doing in developing assisted reproductive technologies for the domestic dog, including the story of the in vitro fertilization. I normally have this slide of why study dogs because I'm often talking to people that study the more traditional laboratory animal species like the mouse. So I have this up to really emphasize the importance of studying reproduction in dogs. So coming from the Smithsonian Conservation Biology Institute, which is part of the National Zoo, I'm particularly interested in the domestic dog as a model for its wild and endangered counterpart, canids. So there are currently 35 species of canids, 5 of which are considered endangered or critically endangered. And these include dogs, wolves, foxes, jackals. And even the species that are not considered endangered are actually under a lot of population pressure, due to mounting threats of loss of habitat or the spread of diseases from domestic dog populations into the wild canid populations. And there's also, of course, a lot of interest in understanding and studying dog reproduction for the purpose of improving things for domestic dogs. So one aspect of this would be developing better contraceptives for population control. We need to understand their reproduction in order to do that. Also, domestic dogs share over 350 genetic diseases in common with humans. Excuse me, which makes them really valuable biomedical research models, but to be able to really optimize their use in that field, we would need to be able to consistently produce individuals with the genes of interest, which would be ideally through transgenesis, which in turn would need us to be able to develop various assisted reproductive technologies. Finally, there's the development of these assisted reproductive technologies, or ARTs, could be used and applied for rare or working breeds of dogs. So one example is actually guiding eye dogs. Most of these dogs that go through the program are spayed before they actually complete their training. So we don't actually know which dogs are the best at being a guiding eye dog until after they've already been spayed or neutered. So if we could actually propagate these individuals by going back and using ARTs, once we know that they are the best guiding eye dogs that we've seen, then we could actually have these best dogs represented in the population. So what would we like to do? As I said, we'd like to control reproduction, so for developing contraceptives or to improve breeding or timing. And the big focus is actually in genetic management. So this can be from rescuing maternal genomes of animals that pass away before they're able to reproduce or for some reason are unable to reproduce, and specifically for better maintaining biomedical research disease models, specific genetic types. So I have this diagram of the ovary up here in the top right-hand corner, and I'm actually going to use my little pen to circle it. So in any ovary, to give a brief overview, you're going to have hundreds of thousands of these primordial stages of ovarian follicles. So this is basically just a single egg or oocyte surrounded by a layer of helper cells or granulosa cells. And these are present in any age ovary and any reproductive stage. There are going to be primordial follicles present. At some point, these follicles are recruited in cohorts are activated to become preantral follicles, where the granulosa cells around them will begin to proliferate and develop. And then the follicles will begin to recruit vasculature. I'm not sure how well this is actually showing up on here, but recruit the vasculature that will allow the follicles to start pulling in liquid, basically water, into the center of the follicle, which will create these little cavities, which are indicated by the white gaps in there. At that point, the follicle is considered early antral because eventually those small cavities will coalesce and become a single large antral cavity. And the fluid in this antral cavity is thought to be extremely important for the development of the egg, so it could actually become able to mature later on and developmentally competent. Eventually then this growing egg will become dominant, and then with the signal will be ovulated, and then what is left of the follicle behind in the ovary will become the corpus luteum that produces the progesterone needed to maintain pregnancy. So this is a brief overview of folliculogenesis. So this is a chart of the various assisted reproductive technologies that have resulted in live births in a variety of species. So this is, again, not all the arts that are out there, obviously, and not all the species, but kind of just a snapshot. So I have the transfer of naturally produced, in vivo produced embryos from a female to a recipient uterus, the culture of these ovarian follicles to result in mature eggs that can be used for in vitro fertilization. In vitro fertilization itself, so bringing the sperm and eggs together in a Petri dish. Very similar to that, intracytoplasmic sperm injection, which is actually the injection of sperm into the egg to facilitate fertilization. And then finally, embryo cryopreservation or freezing. So in the mouse, you can see we've worked out a lot of these technologies, and it kind of goes less so down the line until you see this poor dog highlighted in the yellow, which obviously has less of these technologies have resulted in live births. And I'm going to talk a little bit about why that is, and then also discuss our advancement of adding the one more check mark for in vitro fertilization to this species. So this is the section I like to call why dogs are weird. But basically, why is it that yellow bar, why the dogs have not had as many development of successful arts yet. So one of the aspects is the reproductive cycle. So this is a little bit overwhelming to look at first, but this is an overview of the reproductive cycle of the dog. And I'm actually going to start here, oops, my little thing went away, in the proestrus section. So dogs have about 2 weeks of proestrus, wherein the preovulatory follicles are going through their final stages of growth, and the female begins to exhibit the behavioral and physical signs of what we call heat. And then there's eventually a surge of luteinizing hormone, or LH, which actually stimulates ovulation 2 days later. This is actually considered day 0 of the reproductive cycle. So for the rest of this presentation, when I'm saying day 0, I'm meaning this. Then ovulation occurs 2 days later, and during this time there is a very high amount of progesterone being produced by the ovary, which will last for the duration of the luteal phase, which is this whole section here, regardless of pregnancy status. So following all this, there is a period of what's called an estrus, and this is considered to be a period of ovarian quiescence where there's very little ovarian follicle development happening and no ovulation occurs. And this can last anywhere from 3 to almost 10 months in the domestic dog, and in wild canids that are seasonal breeders, it's consistently the 10-month mark. So basically, there is very little. There are very few times, maybe once or twice a year, where eggs are actually ovulated in the dog. This is something that's a little bit an aside. I don't normally consider this one of the huge issues in developing arts in dogs, but I did want to touch briefly on the anatomy portion too, because it's relevant later. So the domestic dog has a large fat pad covering the ovary. It's called a bursa, and this is, I have a cat here as a comparison so you can see that you can see straight to the ovary, and the cat doesn't have this fat pad, and this has made it difficult to assess follicular dynamics on the ovary in vivo, say if you're doing an ultrasound, because it's hard to see through this large fat pad. And I also want to point out that the oviduct actually runs through this, and I'm going to try to like draw how it is right here on this. It runs through the fat pad, which means it's a little bit more difficult to access the oviduct surgically, and that's where the eggs are going to be those once or twice a year that they are ovulated. So even when these eggs are ovulated, these oocytes are ovulated at an immature stage, so it's the germinal vesicle stage into the oviduct. And they require an additional 2 to 3 days in the oviduct to complete meiosis and become capable to fertilize. And the result of these two aspects means that there are just not that many mature oocytes to use for development of assisted reproductive technologies such as in vitro fertilization. So our goals for genetic management of either laboratory domestic dogs or for endangered canids is going to be outlined here. This is what my colleagues and myself at the Smithsonian and our collaborators around the world are working toward. What we'd like to do is when we have a valuable dog or canid that passes away or that we want to rescue the genetics for, we'd like to be able to take advantage of those primordial follicles that are present in any age ovary and any reproductive stage. We'd like to be able to grow them in vitro until they develop into advanced stages of follicles, where we could then collect oocytes and mature them, mimicking that 2 to 3 day process in the oviduct that matures them in vivo, and then do in vitro fertilization to get embryos, transfer those embryos back into a recipient uterus, and produce live pups. So overall scheme goals. Unfortunately, there's a little bit of a catch-22 in this scenario, because without a lot of mature oocytes available, it's hard to develop in vitro fertilization protocols. The way to get around this would then be to get eggs from the ovary and do culture and in vitro fertilization, and then you could have buckets of oocytes, as my advisors at Cornell would like to say. But you can't develop those protocols either, because without IVF, you don't know if the eggs that you've produced by in vitro fertilization are actually of good quality. So we have this circular issue. And this actually brings us to our work on in vitro fertilization in the domestic dog. And this was a project, as the moderator mentioned, that I did as part of my dissertation. And it was a collaboration between Cornell University, the Baker Institute of Animal Health, and the veterinary hospital there, as well as the Smithsonian Institution. So we had collaborators on both ends working on this. As you can see, there's a lot of people involved in the project. I did show the IVF history in the checkmark table earlier, but I like to show this one because it shows that IVF has been around for a very long time. We're probably mostly familiar with the human story, but even the domestic cat has had successful IVF resulting in live birth since the 1980s. So those issues with the dog are not insignificant. They have actually held back a lot of the advancements in developing assisted reproductive technologies. There have been a lot of IVF studies, though, so it's not just for a lack of trying. The first work in IVF in the domestic dog began in the 1970s, and I've included just here, this is a snapshot, again, of some of the studies in the past 2 decades or so. And in these, researchers took oocytes directly from the ovaries, so meaning that there are various stages of the reproductive cycle and from different stages of follicles. And they matured them in vitro using various protocols and then did in vitro fertilization. So there are thousands of eggs that were used in between these studies. But the rates of embryo production ranged from about 2% to 33%, and none of these resulted in live births. So one of the things when we were assessing IVF is that we thought, well, one of the things that these studies had in common is that they used in vitro matured eggs, so mimicking that 2 to 3 day process that we don't fully understand yet in vitro. And a lot of times the rates of nuclear maturation of these eggs were fairly low to begin with. So it's very likely that the oocytes produced by these in vitro maturation or IVM protocols were not necessarily of the optimal quality to start with to do in vitro fertilization. So there are also a couple studies, though, that used in vivo matured eggs, so waited 2 days or 3 days after the eggs had ovulated to collect them. And in these, you actually had slightly higher rates of embryo production, but if you notice, too, they had much fewer oocytes to work with just because not as many are ovulated. You don't have access to the thousands of eggs in a given ovary. So when we went to look at this, we sat back and wanted to boil it down to really the simple aspects of what could be affecting in vitro fertilization in the dog. So really what it comes down to is that you need to have good sperm, good eggs, and good culture conditions, to put it overly simply. We had an idea from these previous studies that oocyte quality, we could kind of reduce that as an issue by collecting the in vivo matured eggs after they had ovulated and matured in the oviduct. Regarding the sperm quality, we went back to the paper by Mahi and Nagashima in the 1970s that first described the incubation conditions for sperm prior to fertilization, and wanted to reassess that based on some new knowledge that had come out in the past few decades. And finally, we looked at the culture conditions. So specifically, if the culture medium needed to be more simplified, so in the case where maybe there's a component that's inhibiting fertilization, or perhaps more complex culture medium, so the embryos need more amino acids or require antioxidants in vitro in order to improve cleavage. We also explored within this subcategory, I guess, the addition of progesterone to culture, because I had mentioned it was very high in circulation in the dog even prior to ovulation, and so it might have a functional role on the fertilizing egg or sperm. So these are our hypotheses. We thought that using day 5 in vivo matured oocytes would be fertilizable in minimally supplemented medium. We thought that progesterone would improve rates of embryo development. And then going back to that 1970s manuscript, which I'm going to talk about later, we thought that addition of magnesium would improve the sperm's capacitation ability. So with regard to the sperm, so I have a couple slides in here that are listed technique, just because I wasn't sure what the background was for everything. I wanted to give a full scope of the project. But I'm going to kind of skim over most of these, but this is just what we did when we were doing the project. We manually collected sperm from a couple very kind donor dogs, and we then proceeded to capacitate it. So sperm capacitation is defined by this Embryo Project Encyclopedia. I rather like this definition, as the physiological changes in spermatozoa that spermatozoa must undergo in order to have the ability to penetrate and fertilize an egg. And a lot of these changes are related to changes in the membrane of the acrosome, the head of the sperm, that will allow it to bind and actually penetrate, I guess it says, the egg. And that's on the right side of the figure here. Let me get my pen out again. on this section. But it's also characterized by the sperm gaining the ability to, or gaining hyperactive motility. And that actually is shown in this figure down here on the bottom left. So here the tail or flagella changes its movement patterns in a way that helps it better navigate through the viscosity of the female reproductive tract. And this is, again, called hyperactivation. So that the initial description of sperm capacitation medium by Mahi and Nagashima in the 1970s had actually tried magnesium addition because it has been useful in other species. And they ended up omitting it from the final capacitation medium on the basis that magnesium reduced the rates of sperm acrosome reaction, so this right side, after 6 hours of incubation. There just weren't as many sperm undergoing that reaction. However, now we know that it wouldn't be very useful for the sperm to basically lose all the enzymes in the acrosome that they need to penetrate the egg before they actually reach the egg. So if we want to really reassess the role of magnesium, we should do it under the influence of a physiological stimulator. So when the sperm gets to the egg, they hit a lot of hypergesterone, they hit the zona pellucida proteins that are coating the egg, and these will actually stimulate the sperm to exocytose the acrosome and actually be able to enter the egg. So we want to look at it under the guise of a physiological activator. So we started reassessing this for the with regard to hyperactivation first. And so in this graph, I have 4 different treatments. The non-capacitating control, the 0 millimeter here represents the standard canine capacitation media developed in the 1970s, omitting magnesium, and then 2 treatments with magnesium present at 0.1 and 1 millimolar. And here we looked at the percent of sperm that were hyperactivated after 4 hours. And we saw that in the presence of magnesium, there was significantly higher numbers of sperm hyperactivated after 4 hours of incubation. We then looked at the acrosome reaction or acrosome intact status. So here on the right is actually a image of how we evaluated this by staining the acrosome cap. So this is an intact acrosome where you can see the head of the sperm is staining very darkly, and then this is an unintact. So this means the acrosome has reacted and has exocytosed. I don't know exactly if exocytosed is the correct way to use that word. And we use the same treatments, non-capacitating, 0, 0.1, and 1 millimolar of magnesium supplementation. And then we also evaluated control, which actually this is in the black bars. This is the conditions like Mahi and Nagashima had done in the 1970s. And then we also added our physiological stimulator of capacitation, or sorry, of the acrosome reaction. And if we look at just the black bars here, we saw exactly the same thing as they did in the 1970s, where addition of magnesium did not increase the rates of acrosome exocytosis or acrosome reaction. However, in the presence of a physiological stimulator of progesterone, we saw dramatically increased rates of or percentages of acrosome exocytosis in our sperm. So this is great. We have gone back and looked at things based on some new information that we have and found a way to improve our sperm capacitation protocol and work on that portion of the IVF story. So then we switched to eggs. This is critical because when we want to collect in vivo matured eggs, we need to collect them within a very specific time frame. We want them after they're mature, but before they're so old that they're starting to degenerate. And for this, we need very robust estrus monitoring techniques. So we used a variety of things to try to narrow down the day of the LH surge and ovulation. So our first hint was vaginal discharge during proestrus. We then looked at quantification of cells via vaginal histology. I have a slide on that next, but I'm really going to skip through it very fast. We also, more importantly, monitored serum progesterone in these dogs. So this is again that nice overview slide developed by Dr. Kincannon. But you can see that at the point of the LH surge, progesterone is already increasing significantly. My line was terrible there. But progesterone is already increasing significantly in serum. And so we can use these predictable changes in progesterone to time when the LH surge and ovulation occurs. So I have some numbers here, but they're maybe not the same that you would see in some other manuscripts or textbooks, simply because I found that when I switched from the initial report of the study from a chemiluminescent EIA to an RIA, the values I had to use as cutoffs changed a little bit. So it was very consistent within the assay, but something that you might want to double check on using, say, an LH test kit such as this in order to confirm that your ranges are accurate before you start any of this work. So again, that's the vaginal cytology aspect, where they're cornified cells in estrus. So then after we have our day narrowed down specifically, we spayed the dogs and collected oocytes. So to do this, we actually excised the oviduct out of that ovarian fat pad, the bursa, carefully dissected it out, including past the uterotubule junction, so where the oviduct junctions with the uterus here. And then what I ended up doing is usually dissecting the oviduct in half and then cannulating with a 22 gauge needle in both directions and flushing with a warmed saline solution. And this gave a clean cut for which I can get less debris when I was flushing out. But the important thing here too is that within about 24 hours of ovulation, all the oocytes end up around the uterotubule junction area. So this is really the key area, and that's why I usually would cut down into a bit of the uterine horn to make sure I didn't lose any of the oocytes with that. So as I said, we were looking at different days post-LH surge, and for our pilot studies, we're looking at different complexities of media. So I had synthetic oviductal fluid and NCSU, the latter of which was developed for pig embryo culture. And then those are the very simple versions. And then I had a modified NCSU wherein additional essential amino acids were added, and then what I called a complete NCSU or CNCSU that had both essential and non-essential amino acids. And in these, we saw that we had some low levels, but pretty much similar to a lot of those other studies with in vitro matured oocytes, embryo production. And we actually saw decently high rates of what I called delayed cleavage. So this was actually when, normally when we were doing the IVF, we'd collect the oocytes and then do the IVF, and then about the 48-hour mark after collecting them, we would see 4-cell embryos. In the case of delayed cleavage, we actually didn't see embryos until about 72 hours in most cases, and in many of those situations, the embryos we initially saw were 2-cell stage rather than 4, so they didn't immediately go from 2 to 4 like the normally cleaving designated oocytes or embryos had done. So then we looked at day 6 post-LH surge, pushing it further back one more day. And in this case, both the simplified and complex medium compositions were equally adept at allowing us to produce embryos, normally cleaving embryos, although these sample sizes are very small for this pilot study. But based on this, we decided, well, day 6 was probably the way to go, and that also we were going to stick with the complete NCSU just because it did give us more flexibility with day 5 embryos, so we thought it might be a little bit more robust. So this is actually a picture of a single oocyte as it went from fertilization through to embryo development. The dog oocyte is very dark. It's in this right here because it's full of lipids. After fertilization, post-IVF 14 hours, my picture is not blurry because I'm a terrible photographer. It's actually because the sperm have bound to the egg and they are still nodal and they're actually making the oocytes spin. And then after 48 hours, I have well, this looks like a 3-cell, but it's probably a 4-cell embryo because there's probably another blastomere down here underneath that we can't see. And then at the 96-hour mark, we have approximately 8-cell embryo. So when we look at the overall rates of embryo production, again, day 5, we had fairly low rates comparable to what we saw in those previous studies using in vitro matured oocytes. But day 6, we had very, very high rates of embryos being produced, between like 70 and 80%. However, when we look at progesterone versus no progesterone supplementation, we saw no difference in the rates of embryo production in the presence of progesterone. What we did see an effect of, surprisingly, in a way, was the male donor. So this is something that we looked up at kind of at the end. We had looked at the sperm during the capacitation experiments, and there was actually no difference in the capacitation ability of the different males that we had used as sperm donors in the project. So we're a little bit surprised to see such a dramatic effect in some of these with regard to embryo production. The 10-year-old Labrador was perhaps not super surprising since he was a little bit older than the rest of them. However, what was interesting was this 6-year-old Cocker Spaniel male having a very high percentage of embryos that were experiencing delayed cleavage. And I'm pointing him out because it comes into play later, but I'm wondering if the delayed cleavage wasn't necessarily as bad as we thought it was when we were initially observing the embryos, because this male actually had one of the higher rates of live pups out of the number of embryos we had transferred for him. So, because we needed to specifically time our embryo transfers, now that we had all these embryos that we were producing, we needed a way to make sure that our recipients were at the specific time as well. And this is very difficult to do with fresh embryos. So we ended up freezing the embryos using a commercially available kit, which the information is there. But there are also a couple different ways that other researchers have done to freeze dog embryos very successfully. So after we have our frozen embryos stored, we wait for a recipient female to reach the same stage of her reproductive cycle, and then we wanted to transfer the embryos. So if you remember that fat pad around the ovary where the oviduct is going through, this makes it very difficult to get the embryos into the right stage of the reproductive tract. So in the dog, embryos do not actually migrate into the uterine horns until about day 11 post-LH surge. When they're at the 16-cell morula stage. So we had primarily 4-cell embryos, which were day 8 post-LH surge after the whole collection and IVF procedure. So they should be in the oviduct at this time, but it's difficult to get them there. However, some other researchers have successfully been able to transfer 2- and 4-cell embryos into the uterine horns of recipient dogs and have live births. So we started with that since that was going to be a lot more straightforward, less tissue traumatizing procedure to start with. So this is the uterine embryo transfer, basically a stab incision in the cranial portion of the uterine horn and then pipetting the embryos in. Very straightforward. When we did these embryo transfers, we had a variety of fresh, frozen, thawed, and to horns. We sometimes tried doing artificial insemination in case there was some sort of uterine factor that was being missed here that they needed. But in any cases, we did not have any pups born from the IVF procedure when we were doing uterine horn embryo transfers. We did see some implantation sites, some possible heartbeats, but nothing actually resulted on pups in pups on the ground. So our final one, we actually opted to attempt the oviductal transfer. For this, we actually recruited a lovely large breed hound female with nice big oviducts and transferred 19 frozen thawed IVF embryos into her oviducts. So oviductal transfer is a little bit more complicated, I guess. We had to exteriorize the ovarian bursa, open it up to see the infundibulum, and then cannulate that with a Tomcat catheter, which is loaded with our embryos. In the future, I'd be interested in developing this with a laparoscopic technique, because I think it would be possible. But for now, this was the most straightforward way to go about going through the bursa. So in our embryo transfer, we ended up finally with live heartbeats on ultrasound. We had counted initially 6, and with pups with heads and hearts. And we ended up doing a C-section because the female went a little bit past her due date. And I'm not sure if this was necessarily a product of the IVF or the fact that we had transferred IVF for frozen thawed embryos, but since it is an issue in some other species generally that are giving singleton birth, we decided to not try to wait and just did the C-section once she went past her date. And again, this was a large effort between Baker Institute of Animal Health at Cornell and the Cornell Veterinary Hospital, including the very nice Dr. Chung from Theory of Genealogy here, who's waiting to work on removing the umbilical cords from these 2 puppies. We ended up actually with 7 puppies. There was a surprise, and they are all pups were born live and healthy and screaming very loudly. And we then proceeded to do genetic confirmation. So for this, we actually sent out cheek swabs of potential sires, as we had several male sperm donors, all of the oocyte donors, the embryo recipient, out to 2 independent companies for genetic confirmation. And so with our 7 pups, we actually ended up with puppies born from 3 independent IVF procedures involving 3 egg donors and 2 sperm donors. So I like to say it's a nice eclectic family that we had here. And again, this is the 6-year-old Cocker Spaniel here that had the high rates of delayed cleaving embryos after IVF. And we actually only transferred 4 embryos from him into the recipient female. So he actually had a 50% success rate, which is why I think maybe it's not the end of the world that they were a little bit slow. All right, so where are they now? These puppies are now about 1.5 years old, and they've all been adopted out. So this on the top right-hand corner is Cannon, after Dr. Patrick Kincannon, whose graph I had showed earlier in the estrus monitoring. And they're very happy and healthy, and we're actually keeping an eye on them too to make sure that they stay that way, and in part because we wanted to make sure that they are fertile. So we actually waited until they hit puberty before spaying and neutering. And in the 4 males, we had motile sperm in all of the individuals, which was great. And for the females, we actually did the same estrus monitoring and oocyte collection and IVF. So 2nd generation IVF, I guess. And for 2 of the 3 females, we did produce embryos, which are currently frozen, I believe, in liquid nitrogen up at Cornell University. And for the 3rd female, she did not produce embryos. She had ovulated oocytes, and I think that was actually due to poor timing of the spay and oocyte collection. So something that is really a critical component of this whole process right now. So now how do we transfer this technology from domestic dogs and laboratory dogs into breeds of interest or genetic lines of dogs of interest or even the wild endangered canids? So again, this is our graph. We have been able to develop techniques for the in vitro fertilization, and I'm fairly confident, although improvements can be made on our embryo transfer techniques. But we still need to be able to get around the fact that we can't get eggs at any time of the year. And so, pardon. What we can now do that we have a working IVF protocol is work backwards and actually use that to develop a better oocyte maturation protocol, and then working backwards again, a better in vitro follicle culture protocols. So this is what I am working on right now. So starting with the in vitro oocyte maturation, I'm actually presenting a lot of work from some various labs that have been working on this problem for the past couple of years while we've been waiting for IVF to get up and running. And some of it is really cool. Our lab is, my advisor, Dr. Nutran Songsason, had done a lot of work on this in the past, and we're starting it up again. But I wanted to present some of the really cool work that some of these other labs are doing. So this study here, again, I have my overall assessment of what the hormone milieu is like during proestrus and estrus, but they mimicked this in vivo. So Babu et al. Basically added these hormones in sequence mimicking the rise in estradiol, the LH surge with FSH surge, and the increasing progesterone at the time of ovulation in their oocytes, which I thought would be probably a very good way to try to get these oocytes primed and matured. So this day 3 represents the LH surge, and then when they checked everything on day 5, they should have been supposedly ovulated and somewhat matured. And what they found actually was very similar rates of meiosis resumption success, so 2 to 34%. So unfortunately, still fairly low considering all the work put in. The other group I wanted to highlight again, though, there are a lot of groups in Japan and Brazil, and the United States, Korea that are working on dog in vitro oocyte maturation, but is Karine Raynaud's group. And she's done some very cool in vivo studies with oocyte maturation. So for the first one, she took ovarian follicles right around the time of the LH surge and actually collected follicular fluid and then used that to do IVM in oocytes. And what she found was that a lot of the oocytes became degenerate, and the rates of meiosis resumption were pretty similar to what had been shown in other studies. She then went directly to the oviduct and took these oocytes from N. estrus ovaries and transferred them directly into the oviduct of a female around the time of LH surge and ovulation, or I guess 2 days after ovulation, and had, I guess, slightly less degeneration rates, although fewer follicles were recovered, and then again low rates of oocyte maturation. Finally, she collected oviductal fluid by flushing the oviducts with saline and collecting all that, and did in vitro maturation of the oocytes in this fluid. So this is probably a very painstaking process because I'm imagining, I know they don't have very much fluid in their oviducts. So collecting this from dog after dog would take a while to get enough to do this study. And she reported that it helps a bit. So I'm waiting to see the final results from this work. I believe it's going to be published in the next month or so. But it sounds like even when you're basically putting them in the prime area and the oviduct, that it's possible that these oocytes from N. estrus ovaries, from immature follicles, are just not capable of resuming meiosis, at least not at very high rates. So this is kind of interesting, and it helps to inform our stuff. However, it does indicate that oviductal fluid may have some sort of key factor in it if it does help some for the oocyte maturation. So what we're actually doing then, based on that data, are to try to inhibit maturation in our oocytes, give the eggs time to grow and become developmentally competent before we stimulate them to undergo nuclear maturation. So what we started doing is testing various inhibitors of meiosis, so including roscovitin and milrinone, and looking at morphology and then also whether or not they work. And so we're going to start with this and then start adding in some different ways to stimulate egg quality in vitro before the IVM part. So then, if we can, working on this concurrently, we're also working on ovarian culture in the dog, and this is actually some dog follicles from the primordial stage here on the left up to the small antral stage here with that dark oocyte. And the work that I'm doing right now is actually a collaboration between Smithsonian and some biomedical engineers at Stanford University. So there are two different main ways that we can culture ovarian follicles that we use in our lab. The first one is ovarian tissue culture, and this is just taking a strip of the ovary and growing it in vitro. The advantage of this is that the follicles maintain their structural rigidity in the tissue and also the hormonal environment that they're exposed to with all the somatic cells and fellow follicles nearby. Unfortunately, if the follicles are embedded in tissue, we cannot monitor their development over time or response to various gonadotropins or different factors. And then we also found, and I'm going to show this in a second, that there's poor viability of the center of the tissue over long-term culture, and we think it might be related to lack of nutrient availability. So the other alternative is isolated follicle culture. So like I had shown in that slide earlier, where we've basically isolated each individual follicle at various stages, we can do this and then embed them in, say, a three-dimensional scaffold, a hydrogel, in order to try to maintain that structural rigidity that they need. In this case, we can track their development over time. It maintains a three-dimensional structure still, and then the scaffold serves to allow nutrient diffusion and waste removal, unlike maybe the tissue might do. Unfortunately, though, when we isolate primordial stage and primary stage follicles, they really go downhill quickly. They don't like being isolated, basically. And then, as you may have noticed in the previous slide, there's a huge size difference between a primordial follicle and a preovulatory. Well, even this is the small antral follicle, not even a preovulatory follicle. But this is an enormous size change. And so this requires maybe a very dynamic culture system to support the changing needs of the follicle as it makes this huge change. So this is some work done by a previous postdoc in our lab, looking at some different ways to culture ovarian tissue. So she tried to control basically with the tissue in culture medium, growing the tissue on a cell culture insert, and then also growing it on an agarose gel block, which has been successfully used to culture testicular tissue. And what she found, and this is comparing cat and dog again because it gives you a sense of dogs being difficult, that after about 3 days of culture, the agarose gel was really the best system for maintaining follicle viability in both species. However, it didn't necessarily last for long-term culture, so we still need some things to work out. This is a picture on the top left-hand corner of the agarose gel blocks with pieces of tissue growing on them. And then this is what I was talking about the center of the tissue becoming degenerate over the course of culture. So the upper right-hand corner image is a calcinethidine bromide stain where the green represents live cells and the red dead cells. And what you can see is that center of the tissue has stained red after about 2 weeks of culture. The bottom left-hand corner is the similar thing where you can see there's a odd patterning. You don't see any follicles on the bottom right of that in the center of the tissue, but there are follicles on the outer edge. So the same postdoc, Dr. Fujihara, also cultured some ovarian tissue in a bioreactor that had continuous perfusion of medium through the tissues over culture. And in that case, you actually saw much higher percentage of viable tissue, center tissue, after the same amount of culture. So that's the bottom right-hand picture compared to the top right-hand. So doing this, we wanted to develop a system where we could culture ovarian tissue and isolated ovarian follicles under flow. And with this, we were working with Dr. Utkan Demirci's lab at Stanford University to develop what we're calling an ovary on a chip. So we're taking advantage of microfluidic technology to do this. And so basically what we have is 3 chambers, channels in this chip, each with an inlet and outlet hole on the top that's connected to tubing, and we can actually pump medium through the system, continually perfusing our tissue or follicles. And when we did this with domestic dog ovaries, we compared, so then fresh tissue compared to tissue cultured for 3 days on our agarose gel control, and then in the chip system with 2 different rates of flow. So the reason we're looking at different rates is that we don't want it to flow so slowly that it doesn't really have the benefits of continuous perfusion or flow. It doesn't get enough nutrients to it, and it also can't be so fast that you're basically blasting the cells and causing shear damage to them, or potentially even washing away the paracrine factors that they want to continue to develop. So looking at this, we have all these little primordial follicles in the tissue, and what we found when we went back and looked at the proportion of normal morphology follicles over all the follicles per millimeter squared of tissue is that dog follicles did not really like culture under flow, at least under these conditions. And with the increased flow rate being particularly detrimental, and particularly to the primary stage, the activated follicles. So that's not ideal. And then we also looked at the diameter of the follicles in the system. So like, okay, maybe the follicles, there aren't as many follicles in the tissue, but they are bigger, they're growing. But we didn't actually see a particularly increase in primordial follicle sizes in our tissues. In fact, in the high flow rate, there was a decreased diameter of these follicles, indicating that there was possibly a shear effect in that higher flow rate for the dog. So just as a comparison, I have a slide also for the cat. We usually do a lot of comparative ones to see what's going on. And in these, the populations of primary and primordial follicles actually were maintained similarly to the agarose gel controls in our ovary on the chip microfluidic system, and there was no additional effect of increased flow rate on either way on the follicle populations. So we also did isolated follicles. So this is actually an image of some primordial primary stage follicles, and then we embedded these actually in a hydrogel at the base of the channel, and then made sure that media could perfuse down into that layer where the follicles were embedded, and then did flow versus static in our same channels. And so far, in using preantral stage follicles, so primordial through secondary stage, we found that there was no significant influence of flow on isolated follicle growth. However, right now I'm working on, like today, some preantral and early antral stage isolated follicles cultured in the same way, and they appear to be doing a lot better under the flow. So that might make sense because around the early antral stage they become vascularized and would be expecting to have greater nutrient availability and like exposure to flow. So this may be something that would be more viable for later stages of ovarian follicles, which is what we're looking at now. So the applications for this ovarian chip, as you guys were well aware, I'm interested in the wildlife conservation impacts where we could rescue maternal genomes. Again, also being able to take advantage of genetic management of, say, laboratory rare genotypes from laboratory animals. There's also an interest in this for human fertility preservation because we need a way to culture ovarian follicles in large mammals, and that's just not been successful yet beyond in the mouse. This could also be used to assess the role of environmental toxicants on the ovary in a more natural, I guess, biomimetic environment, which would help reduce the number of animals used in research. And then, in general, understanding ovarian follicle development from the scientific standpoint, possibly to develop, say, contraceptives. So I'm going to wrap it up there. I have a lot of people to thank from a lot of different labs. For the first, I'd like to thank my dissertation family at Cornell University, the Baker Institute for Animal Health. Their animal care staff were fantastic at helping us with all these things and taking great care of all the puppies. The veterinary staff, the surgeons and veterinarians that helped us with IVF puppies. And then I have a lot of students that also helped with the project that are pictured here. And then also my current lab at the Smithsonian Conservation Biology Institute with my advisor, Dr. Nutrin Songson. Oh, and I didn't mention my advisor at Cornell, Dr. Alex Travis, of course, and SCBI scientists and staff, and then our collaborators at Stanford University helping with the Ovary on the Chip project. So with that, I would like to take any questions. Thank you so much.
Well, thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question for you, Doctor, is, with regards to the chip and follicle culture, what do you think is a reason for the differences you saw between dog and cat tissues?
Hi, yeah. So our thought with regard to the difference between dog and cat is actually we're working on teasing that apart a little bit more. But what I'm thinking actually is that it's due to a difference in rigidity of between dog and cat tissue. So if anyone's had experience cutting through an ovary of a dog versus a cat, the cat is like, it's like cutting through, maybe I use food analogies all the time, so pardon me for it, but like a ripe pear or something like that, versus cutting through dog tissue can be upwards of like cutting through an apple. It's much tougher, and so I think maybe that this tissue is more fibrous, or certainly more rigid. So that is possibly the reason why it's not responding as well, particularly in ovarian tissue culture, for follicle growth. It may be that rigidity has been messed up somehow.
Our next question is, are there any particular health issues or concerns you're watching out for in the IVF pups as they grow?
Hi, yes, actually we are monitoring them as they grow. As I said, they're only about a year and a half now, so they're still fairly young. But a lot of evidence, or evidence is starting to come out more and more, particularly with regard to human-assisted reproductive technologies, that some of the technologies have resulted in metabolic disorders, particularly in children. So we're going to be watching out for those as the dogs grow and develop.
Zoran, looks like we have time for one last question here. This question is, how long would it take to complete the process of genome rescue as you have proposed, for example, immature follicle culture through the birth of live pups?
That's a good question. So it's a little bit complicated to answer. So we think that the growth of follicles from the primordial stage through ovulation in the dog takes about 100 days, which would be similar to, say, a human. And then you'd need to then do your oocyte maturation, in vitro fertilization, and then transfer embryos to a recipient for a 2-month gestation. So this could take upwards of what's like 170 days, possibly more if you had to wait for a recipient uterus to be in the right time. So you'd have to like freeze them for a few months and wait. However, there is some research, particularly in human ovarian culture and a couple other species, where there might be a way to accelerate the follicle culture process. So, for example, this is again on testes, but when they've xenografted testicular tissue into mice, they have found that what would take normally, say, 15-week time frame for the immature tissue to develop and produce mature sperm, when it was transplanted, it only took 5 weeks. So either through xenografting or through culture systems, there may be a way to accelerate this. And so in that case, then that 100-day time frame might be shortened, but upwards of maybe 170 days.
Well, before we conclude, Doctor, do you have any final comments before we let you go?
No, I'd just like to thank you all for allowing me to present my work to you, and thank you to LabRoots for hosting this.
Thank you again, Doctor, for that outstanding presentation, and thank you again to LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. And until next time, goodbye.